afternoon, everybody. At the outset, let me say that I have no commercial interests. I do research at the Ravi Prasada Institute, and unlike a clinician, I don't deal in sponsorships or any companies other than purchasing chemicals, so I have no commercial relationships whatsoever. And I thank Jerry for inviting me to be a part of this course. Once again, it's a pleasure. What I just want to do is just give a short background about genetics briefly, and then I will talk about the diseases that I mentioned. So basically, when we say genetic disorders, we have the so-called, now it's so-called because concepts have been rapidly changing over the last several years, single gene or Mendelian disorders, because they obey Mendel's laws of transmission. And so which basically is that you have alteration in one gene which is necessary and mostly but not always sufficient to cause the disease. And you have the inheritance, which the classically described modes of inheritance as per Mendelian laws, which are autosomal dominant, autosomal recessive, and X-linked. Then apart from that, you have what are called as complex diseases, which have actually become more, I should say, omnipresent. They are the ones that are more common, and they have been recognized now with more newer tools and technologies to actually tease out all the different causative factors. So the very common, like the previous speakers have mentioned, like diabetic complications and heart disease and macular degeneration, age-related macular degeneration, and adult glaucoma, etc., are complex diseases. So you have one or more genes plus environment. So all these factors together contribute to the etiology of the disease. So for Mendelian disorders, we have a mutation in the gene, which is at the DNA level, which gives rise to mutation in the messenger RNA, which is transcribed from this particular gene. That again gets translated into mutant or abnormal protein, which gives rise to the disease. So this is a very simplified schema that actually tells you what the chain is of causation. In complex disease, you have genes plus environment, and they're called as multifactorial and therefore complex, which means many factors. So as I said, you have both of the categories of etiologic agents. In genetic components, one or more genes are involved. You may have familial transmission that is seen fairly often, but the pattern is variable. You don't have very set patterns like in the case of the autosomal and the sex-linked Mendelian types. Genes increase the chance of having the disease, so they don't have a one-to-one. For example, you have retinopathy of prematurity, which is again another example of this class of diseases. So what do we look at the genetics of ocular diseases for? Because they tell you how and why the disease happens. If you want to go down to the basics, this in turn maybe can help you in the longer run. This is a long-term outcome which can lead to new treatments. You shorter term, what a lot of basically genetic testing, which leads to counseling, risk prediction, better diagnosis and management in certain cases. You may be able to correlate the genotypes or the mutation, that the patient has with the phenotype, and that may help you again in terms of the classifying the disease and so on. So coming to retinoblastoma, this is actually a very classic disease, and it turns out to be a model both for oncogenesis as well as for genetic disorders. In many ways, it's a first kind of prototype. So what it is, it's a malignancy of the retinal precursor cells, and you have inactivation or mutation basically of the retinoblastoma 1 or RB1 gene, which is located on chromosome 13, and this gene was discovered in the late 1980s. Can be hereditary or non-hereditary, and usually the hereditary form is inherited in an autosomal dominant fashion, and the penetrance is quite high. That means those who carry the mutation will most likely get the disease, a very high chance of getting the disease. And both alleles of the gene need to be inactivated for the development of the malignancy. So you normally have 2 copies of every gene or 2 alleles of every gene in the genome, and so you need actually both the alleles to be inactivated for the tumor to manifest. So this is again just to show you the first allele is mutated and is transmitted through the germline, therefore it's present in all cells of the patient. It gives rise to predisposition to a tumor. And this has to be accompanied by a somatic mutation of the second allele, so that happens somatically in the retinal precursor cells, and that gives rise to hereditary disease. Whereas if you have the first and the second allele both being somatic, then you have non-hereditary disease. So that means both the mutations are only present in the cells in the retina. So just to again summarize, this is mostly bilateral. It can be unilateral. In hereditary retinoblastoma, you have earlier onset, higher risk of second malignancies. mostly sporadic, although you have 5% of familial cases, whereas non-hereditary you have always unilateral disease, somewhat later onset than the bilateral or hereditary form, no risk of second malignancies like in the hereditary case retinoblastoma, and it's entirely sporadic. Obviously you don't have familial transmission because the two events have been somatic and therefore not passed on from previous generations. And you have bilateral to unilateral ratio generally being 40 to 60, but 10 to 12% of unilateral retinoblastomas can be hereditary. Generally this is the percentage that you have hereditary disease in case of unilateral. So you have sometimes what's known as incomplete penetrance. So this is basically, it means that if you carry a mutation, you have less than 100% chance of getting the disease. So here you can see a pedigree, and the different circles are those who are affected with the disease, the blank circles are those who are normal. So you can see here that two of the children are affected, and if it's dominant disease you would expect that a parent should certainly be affected, one of the parents, but in fact none are affected. This is what we mean by low penetrance. But obviously one of the parents has a mutant gene, so what happens is the risk of the relatives of these affected persons to get the disease is lower than when it has full penetrance or 100% penetrance. And you can have what's called as variable expressivity when you have this kind of incomplete penetrance. So you have this half-filled circles here, which means you have patients with unilateral disease. So in RB, people have found these situations where both of these phenomena occur together. Then these are the kinds of pedigrees for the reasons I mentioned, and also the fact that you have mosaicism. So this is another genetic phenomenon where a person doesn't carry the mutation in all the cells of the body. It only occurs in certain parts of the body. And so depending on whether it affects the retina or not, that person is likely to have the disease. So you can see. So what happens to the pedigree is you have again affected people only in one generation. So this is again a situation where the mutation has occurred post-zygotically. So it's only the present in the affected offspring and not present in the parents. So this is the strategy in general for genetic analysis and counseling. Relatives who may be at risks for familial and non-familial RB would have different levels of risk. So in familial case, if a patient has bilateral RB, the children of that patient would have 50% chance. Depends, of course, assuming it's full penetrance, siblings would again have 50% chance. If it's unilateral, since only about roughly 10% of cases are hereditary, you may have 10 to 50%. Again, it depends on the penetrance, but it could generally be about 10%. In non-familial, again, if the patient has bilateral RB, since this is transmissible as hereditary disease, the offspring would have the same as, in this case, 50%. The siblings would have 4 to 5%. And this is because of mosaicism, because there's 10% mosaicism. And in unilateral it's negligible, so it's just about 1% risk. Right, so that's basically. So the strategy for genetic analysis and counseling, you need when you need actual risk assessment, then there is a cost versus benefit issue, so identification of those at risk can be followed by surveillance of those people. You can avoid unnecessary surveillance and therefore save costs in terms of those who are definitely not mutation carriers. So this is a report that happened by now many years ago, where in fact genetic testing for RB was used for pre-implantation genetic diagnosis, and this was in Dr. Abramson's lab. So this is just to show you the RB1 gene. So this is on chromosome 13, and so this is basically how the gene is organized. You can see it has a large number of exons, which makes it rather involved to screen for mutations, because you have to screen all these different fragments separately, and then you also have to look for multi-exon deletions. So it's really different levels of screening, different levels of testing, and so on, and it's quite an involved process, a sophisticated set of tests that you have to do. And this is the esterase gene, which is was used as a marker for RB1 gene deletions because it's located right next to this. So you have a lot of different types of mutations. You have large and small mutations spread throughout the gene in retinoblastoma, and these are the rough percentages. So the majority of patients have small mutations, which may be a single base or just 2 or 3 bases. Then you also have epigenetic changes in tumors, but that's really not something that you can test as far as the patients is concerned using blood samples. And a few of them have cytogenetic level changes. So you can actually do conventional cytogenetics or molecular cytogenetics, do chromosome tests to find out the genetic alteration. So this is just to picturize the same thing. You have a chromosome, and then this is the level at which cytogenetics and FISH works at the level of the whole chromosomes. You can detect it in 5% of patients. 10 to 20% of patients, you need PCR-based tests because it involves exons of the gene and multiple exons probably. So you need tests like quantitative PCR. And then if you go within exons to single base level, you need PCR and DNA sequencing, and this is like on the scale of 1 to 100 bases. This is the level of mostly of kilo bases, and this is in mils of base pairs or megabase bases. So the majority of patients actually need a single base level screen. So I think people have talked about retinal dystrophy, so I'm not going to go too much into this, but so it's extremely heterogeneous group of disorders, and you have rod cone type, cone rod. These are the examples. You can have color blindness, macular degeneration. You have what's called as a generalized photoreceptor dystrophies, which examples are Leber, congenital amaurosis, Schubert, etc. You have syndromic forms and non-syndromic, and then they are Mendelian disorders with over 200 genes known so far. So this is a graph which is from a database which actually gives you a catalog of all the retinal disease genes discovered till date. It's very, very useful, especially for people who are doing laboratory genetic work in this area. So as of 2013, you can see that it is well over 200, the number of genes that have been mapped, and almost all of these have been identified. So in terms of genetic testing for mutation, for retinal dystrophies, potentially you can have it for diagnostic confirmation and for counseling or prediction. You can determine genes that are frequent in a particular population because you want to, try to develop therapies for those genes preferentially. Or you can do genetic testing to recruit patients in gene therapy. For example, the RP65 gene therapy trials have been ongoing now by several groups in the US and UK. So that would be another reason to actually test patients in terms of for genetic mutations. And these are roughly, this is a pie chart taken from a paper published a few years ago, and this shows you the different genes in LCA and the rough frequency. So you can see that a large chunk of them are still unknown. And then you have all these genes. So there are again many different genes. You have the mutation frequencies here. So again, this would vary somewhat from population to population, and these have been described from the West. I think we don't have a very comprehensive database here as yet, but that's ongoing, and hopefully we should have it from soon from Indian patients. So this just gives you the kind of broad classification of phenotypes based on different gene mutations. So there's a guanylate cyclase gene, and there's GRIP1. So if you have mutations, patients with mutations, any of these would preferentially or largely are present with these kind of features. They would have photophobia, or these are the kinds of features that they would present with. Whereas if they have night blindness, then you have it's highly likely that the patients have mutations in other genes such as LCA5, RP65, RDH12, CRUMS1, and so on. And again, here you have certain more specific features that have been described. Now, these features have been described by really looking at large groups of patients after determining the mutations. So they have looked at groups of patients with mutations in a particular gene, looked at the clinical features, and then tried to derive associations between clinical features and a specific group of patients having mutations in the same gene. So these are basically this just shows you some of these associations. So there are variations in phenotype from one gene to another. Again, it depends on the stage that you're looking at. In more advanced stages, the disease probably, because it's progressive in nature, it may become that the boundaries are not so easy to distinguish. So you have high throughput technology, since you have these group of disorders with very many genes to screen. You have what's called as DNA chips or microarrays, where you can have these spots which really represent individual small fragments of DNA that you can put your test sample on and then pick up by scanning to see whether there's a particular mutation or not. So it's obviously much faster, and you can screen multiple variants or genes, and it's cost effective when you have a large number of tests to do, as in this case of group of diseases. So you have chips that have been developed, and these are just examples. There's an Asper ophthalmics that was also mentioned. So you have LCA more than 600 disease-associated variants, which have already been made into a chip that can be purchased and used. You have a retina array that's been developed, which has sequences of 93 genes that have been involved, known to be involved in retinal diseases. You have the ABCR, this is Stargardt disease gene. So in this, again, is a large gene with a large number of mutations. So you have about 400 disease-associated variants which have been coded on this chip. And again, you have other chips that you can use to map the disease locus. So the most recent technology is the acceleration sequencing, where you can sequence the entire genome or the coding genome of a patient, the exome. You have nowadays, offers of personal genome sequences that are being offered. You have, it's powerful because obviously you can detect rare genetic variants even with single patients, and it reduces the time and effort. So if you, instead of screening 200 genes, you can simply screen the whole genome, pick up the whole lot of variants, and sift through them to identify which the variant is that is relevant to the disease that you are interested in. And it has led to the identification also of new genes for retinal diseases. So even from a research point of view, it's useful, and that's all. Thank you.